Hi, this is Seher from Easy Peasy, and today we are going to discuss enzymes. Enzyme is a really big topic, okay? We have a lot of different terminologies that's going to come, and they are really close to each other but have differences in each other. So I will recommend you guys to have a paper and a pen with you so that you can notice the difference. And I also put a 5-second quiz in the end so you can analyze at how much you get from this video. If you think you didn't did much in that quiz, watch the video again and then take the quiz again, okay? And hopefully you will get what I'm trying to say, and I'll try my best to give you as much clear and easy way to remember all those terminologies. Okay, now let's start. Fine, so enzymes are protein catalysts that increases the rate of reactions without being changed in the overall process. So they're not going to get involved in the process, they're just a helper. They're just helping to increase the rate. Now, enzyme names are derived from the substrate or the chemical reaction the enzyme catalyzes. So if I say that we have a reaction that says transferase, it means something is transferring from one to another place, right? And if I use the word oxidoreductase, then it means I'm talking about oxidation or reduction here. So the word ase is basically giving me the idea that I'm talking about an enzyme. Fine? Okay, next. Next is nomenclature. So we have two different categories in it. One is recommended name, another one is systematic name. Recommended name like trypsin and pepsin kind of stuff. Systematic name is further categorized into 7 different subunits. Okay, so this is oxidoreductase, transferase, hydrolase, lyases, isomerases, ligases, and translocases. Okay, now oxidoreductases. By the word, you can say it's a kind of oxidation and reduction process. So electron is transferring from one molecule to the other, oxidizing one and reducing the other. Fine? Okay. If I'm using the word transferases, it means I'm transferring something. The example I take here is phosphotransferases. It means I'm transferring phosphate here from one molecule to the other. If I'm using the word hydrolases, lases and lyases means cutting something. And hydro means we are using water for that. So if we have a compound and we want to break it, we are using water there, then this category is going to fall in hydrolases. Cool? Okay, the example here is nucleases and proteases. Next is lyases. So I already told you lyases means cutting the bond. So all those reactions that doesn't fall in the category of hydrolases and oxidoreductases is going to fall in this category, that is lyases. Okay. Next is isomerases. It comes from the word isomer. It's telling you that some rearrangement is occurring there. So example is in front of you. If you can see that there is some rearrangement in the reaction, that reaction is going to fall in the isomerases then. Okay, next is ligases. Now the word gases is telling you that G stands for glue as well. So I'm gluing something here. So two molecules can combine with each other. This ligase is very much similar to lyases. So the difference between the two is G. If you are sounding G there, like gases, so you're gluing it. If it doesn't sound G, it's lyases. It means you're cutting it. Fine. The last category we have here is called translocases. It means that if you're transferring something through a membrane, then it's going to fall in the category of translocases. Fine? Okay. Next, the word that is really similar with each other is synthase and synthetase. Now, these two enzymes have a similar kind of function there, but the only difference here is synthase needs ATP to work, while synthetase doesn't need ATP to work. Now, how to memorize it? It's quite similar with each other, right? But there are two different terminologies. If I tell you one needs ATP and one not, after one day, 2 days, maybe a week, you will forget it, right? You will not remember this thing. So how to memorize it to differentiate between the two, that who uses ATP and who don't? Just see. There are 2 T's present in synthetase. So if you have 2 T's there, it means you're turning down ATP. Just like in the movie of Frozen, Elsa turned down Anna, and in this picture, Anna is complaining about it, right? So turn down ATP means 2 T's should be there. That's going to give you the idea which compound or which word does not use ATP and which word use ATP. Bingo, right? Okay, next. The next confusing terminology here is phosphatase and phosphorylase. Okay, now phosphorylase is basically adding phosphate group, and phosphatase here is removing the phosphate group from there, right? I'm not going to go into details about it. Maybe some phosphorylase are also going to remove the phosphate group from the molecule. It's that word kinase here that is adding the phosphate. So I'm not going into detail about that. The only difference here that I can see is phosphorylase is using ATP molecule, okay? And phosphatase is using water molecule. So how can you remember that, right? 2 things, almost similar function, almost sound similar there, and I need to remember that which one is using ATP and which one is not. Focus on the word R. Phosphorylase. It means you're releasing energy. Just like when Elsa is pissed off, it says enough, and she releases energy there. So phosphorylase need ATP, needs energy there. Easy, right? Okay. Next is dehydrogenase and oxidase. Now these 2 terminologies are quite different from each other. Let's see how they are similar. Now I have 2 reactions here. In one reaction I'm using the enzyme dehydrogenase. In the other reaction, I'm using the enzyme oxidase. The product is similar, uric acid is there, but the byproduct is different. The word oxidase is telling you we are involving oxygen molecule there, so that's easy, right? So oxygen molecule is there. And the dehydrogenase is also telling you that you're removing hydrogen atom and you're removing it from the molecule that does have the atom there. So NADH will be made in the product. Okay, so that's quite easy to remember. But the difficulty lies here. The word oxidase and the word oxygenase. Now see, both words giving you the idea that oxygen is involved, and we need to find out the difference, right? So let's see. In oxidase, we are using oxygen and we are proceeding with the function, and you can see the oxygen comes here in the byproduct. It doesn't remain here with the substrate, okay? But in oxygenase, the oxygen gets incorporated in the substrate here, so you can see this thing is present here. Okay, that's the only difference. Now how will you remember it, right? Just focus on the word genase. Genase give you the idea of the word genes. So it means something is incorporating here, right, in genes. So that's giving you the idea or reference that oxygen molecule is gonna get incorporated in the substance. Cool, right? Okay, next is allozymes and isozymes. Now, allozymes, it differs structurally but not functionally, derived from different alleles of the same gene. Now, if you have a chromosome, the chromosome have 2 alleles in it. It can have multiple alleles in there, right? So multiple alleles is going to produce multiple proteins, but they are going to function same. So here you can see there are different eyes. The color is different, but the function is to see the whole picture. So they all can see, right? But only the color is different. So here they are doing a same function, but they are structurally different. In case of isozyme, it differs in amino acid sequence, but catalyze the same chemical reaction. So again, you have the same function, but in this case, you have different chromosomes as well. So each chromosome is creating a different protein there, and different proteins are performing the same function. So that's the difference between allozymes and isozymes, okay? Next, you know, we always, whenever we discuss the enzymes, we always discuss the model, the old model and the new model. So I'm also going to put this thing here, because if you are really new to the enzymes, you should know that there are two different models here. One model is called lock and key model, and one model is called induce fit model. Lock and key model is each lock has a specific key here. So lock is depicting here as an enzyme, and key is behaving as a substrate. So a specific substrate is gonna unlock or react with the enzyme here, and if it's correct, then they will convert into the product. If they're not correct, then there will be no reaction. Fine. The latest model is induced fit model. It says that the enzyme here, they have a specific cleft called active site. The substrates get fixed into it. It takes a little bit, what you can say, a configuration to accommodate the substrate there. And this accommodation here, or complex here, is called enzyme substrate complex. And then it's going to convert into the product there. So if that's the thing, that if the enzyme is changing its shape a little bit to accommodate the substrate, then we can say it's an induced fit model. Fine? Okay, next. Next is allosteric enzymes. Why I'm using the word allosteric enzymes? Because all enzymes are not allosteric. There is a few category that have the allosteric enzyme. So I will differentiate it right now. I'm just giving the overall view. Okay, so this is the enzyme. This specific cleft is telling you that this is the active site, and this is the allosteric site for this enzyme. Okay. Now, if the enzyme doesn't have substrate in it and it's empty, then we call it apoenzyme. Okay, apoenzyme, apo is giving you the word empty here. So just say that this enzyme is empty right now. Okay, now we are adding a small molecule in it. This is not a substrate. It's a non-protein portion, and it helps the enzyme to function. If it's a small organic molecule, then we call it co-enzyme. If it's not a small organic molecule, it's just an ion like zinc ion or ferrous ion, then we call it cofactor. Okay? Now with this non-protein portion, we are adding the substrate here, and this is our substrate. So these two things are going to combine there like this, and now this enzyme is full, it's heavy, and we call it hollow enzyme. Okay, see, a lot of different terminologies, right? That's why I ask you to have the paper and pen there. Okay, so apoenzyme is giving you the feeling of empty enzyme, and hollow enzyme is giving you the word that the whole enzyme is there, so it is full. Fine? Okay, now if you have allosteric site, like if this enzyme is an allosteric enzyme, we do have an allosteric compound that can attach to that site, right? If that allosteric compound is positive, then it is going to give the positive effect there. And if it's negative, then we call it inhibitors. Fine, so it gives you the idea. So allosteric site, the compound that's going to attach to it can give both. It can boost the reaction, it can inhibit the reaction, right? Okay. Now, types of effectors. So we have two different things. So just say these four different subunits here are basically four enzymes here. Okay, just like we discussed in hemoglobin, we have four different subunits there. if one of the subunits is attached with its substrate, if I'm talking about hemoglobin, just think oxygen molecule is attached to one of the substrate. Remember the heme-heme interaction I discussed in that video? One oxygen molecule is boosting the affinity of another molecule, oxygen molecule, to attach to it. So here the substrate itself is giving the positive effector kind of thing here, right? So if that's the case, then this effector is called homotropic effector. Fine. But if we have an enzyme or a molecule and the allosteric site is different and there's a different molecule that is boosting the reaction or inhibiting the reaction, then this kind of effector is heterotropic effector. Okay? Homotropic effector means substrate itself is boosting the reaction. Heterotropic means that we have another molecule that is boosting or inhibiting the reaction. Fine? Okay. Next is inhibitors. Any substance that can decrease the velocity of enzyme catalyzed reaction is called an inhibitor. So we have two different types here, reversible inhibitors and irreversible inhibitors. Reversible inhibitors are further classified into competitive inhibitors and non-competitive inhibitors. Okay? And irreversible inhibitors are bind to the enzyme covalently. So they are also referred as suicide inhibitors. Okay? Okay, so now we're going to discuss the reversible one first and irreversible later on. So here you can see two different things. One is competitive and one is non-competitive. So in competitive inhibitors, you can see that this inhibitor is taking the place of substrate, and substrate cannot attach to the active site and it's going away. So it does get affected by increasing the concentration of substrate. So if you have a lot of substrate here, this inhibitor is going to replace by the substrate again because the substrate's affinity is more than the inhibitor here due to its more concentration. Fine? Okay. The second type we have here is non-competitive inhibitor. Here, the inhibitor is attaching to a different site. It is not attaching to the active site here. So, in this case, if you increase the concentration of substrate, it doesn't affect the affinity for the inhibitor. Like, it doesn't affect the inhibitor there because inhibitor is attaching to another site. They are not linked with each other. Okay? Next is irreversible inhibitors, right? So I'll take the example here of penicillin. The penicillin is basically inhibiting the formation of cell wall in bacteria. That's how bacteria cannot multiply and they die and we survive, right? So that's the kind of example that can fit into the suicide inhibitors that we talk about. Okay, next is the mechanism of enzymes. So look in the picture, we have the reactant, and then this reactant needs a lot of amount of energy to precede the reaction or to convert into the product. And this energy is called activation energy. Okay, so in this picture you can see we don't have any enzyme, so this reactant needs this much of activation energy to convert into the product. But if we have enzyme here, enzyme can lower this activation energy and the reactant will transform into the product more early as compared to the reaction without enzyme. Okay, so that's the difference and that's the mechanism. Now, how I fantasize the mechanism of enzyme? I just think that the Elsa from Frozen 1 is basically my substrate, and in Frozen 2 she needs to pass the ocean, and the ocean was brutal there, so that's the activation energy you can see. And after passing that activation energy, she converted into the white queen there. Oh my god, I love her. Don't get distracted. Okay, come back. So she converted into the product. Okay, now in this movie she took the help from the water spirit, and that water spirit is basically giving her a reroute and took her there early. So the water spirit is acting as an enzyme here. Okay, now in this picture, why I put this picture here, because I want to show you the overall energy release during the reaction without enzyme and with enzyme is the same. So the overall energy release is not changed. They are only changing the activation energy of the enzymes or the reaction. Okay, fine. So this is the enzyme we have here. Alpha is a substrate on the spirit, ongoing and converting into the white queen. So bingo, right? You know the mechanism of enzyme. Just watch Frozen 2. Okay, so I just give a free, what you can say, publicity for the Frozen 2 here. Okay, next. Factors affecting reaction velocity. So we have 3 different factors that can affect the reaction velocity. One is substrate concentration, temperature, and pH. Now, substrate concentration. Look in the picture, okay? We have few enzymes and we have few substrates. So if we have more enzymes and less substrate, then few of the enzymes will remain empty here. Okay, so if you increase the concentration of the substrate here, all the enzymes can get fixed with or make the complex here of enzyme substrate complex, and it gives the boost to the reaction. But once all the enzyme substrate complex are made and we don't have any more enzyme here to make more complexes, then the reaction comes to a smooth level, and this level is basically the Vmax we have here. Okay, so that's the maximum velocity a reaction can have. Now this is the teeny tiny, what you can say, equation that can tell you the velocity of the reaction depending on the concentration of substrate there. Okay, next is temperature. Now all the enzymes that are present in the body have an optimal rate, so that rate can differentiate from 30 to 45 around. Okay, after that the enzymes start denaturing itself. So if you increase the temperature, that's good. Just like Olaf loves summer, right? So if you increase the temperature, it increases the rate of the velocity of the reaction or the rate of the reaction. But if it increases a temperature more than that, then Olaf is also going to melt. So just like Olaf, the enzymes are going to get denatured. Fine. I think enzyme is based on Frozen, so yeah. Okay, next. Next is pH. Now for pH, there is no specific statement for it. Each enzyme has their own characteristics. So if they are present in our stomach, they are doing their maximum function in acidic environment. If they are present in a normal environment, and they like salivary amylase, then they will function at a normal pH. And the enzyme that works in the basic environment, like arginase, then if you provide them more basic environment, they will function best there. So each enzyme have a specific pH for it. There is no specific statement for overall enzymes here. Okay, next is enzymes in clinical diagnosis. Okay, now there are only a small group of enzymes that actively secreted in the blood. Okay, but most of the enzymes work within the cell. And when the cell goes through the process of apoptosis, then those enzymes gonna get released into the blood or blood plasma. Fine, but there's a process. So enzymes are secreted by apoptosis into the blood. There's also a process to remove it, so it balances the concentration of those enzymes in our body. Okay, now for example, you are ill and you have some kind of disease there. Okay, that infection is basically bursting your cells there. So if your cells are bursting or going into the process of necrosis there, all the enzyme that should be present in the cell is present in your blood plasma now. Okay, now if we check the blood plasma level of enzymes, and if they are more than a certain level, then it tells us that, okay, this person is ill. Okay, and if you have a specific enzyme that works in a specific environment, just like I took alanine aminotransferase, it is present abundantly in liver. So if this enzyme is present in your plasma, it gives you the signal that something is wrong with your hepatic tissue, or there's a damage in the hepatic tissue there. Sounds good? Okay, so that's the overall thing for enzymes. Let's start the quiz. Are you ready for this? Okay, let's see. Fine. My first question is, what is the difference between synthase and synthetase? 5, 4, 3, 2, 1. Okay, synthetase don't need ADP and synthetase do. You remember the two T's there and turn down effect? Okay. Next question. What is the difference between phosphatase and phosphorylase? 5, 4, 3, 2, 1. Phosphatase don't need ATP, while phosphorylase do. Remember the sound of R that releases the energy, like Elsa says, enough? Okay, next. What is the difference between oxidase and oxygenase? 5, 4, 3, 2, 1. Oxidase do not incorporate oxygen molecule in the product, while oxygenase do. Next, what is the difference between allozymes and isozymes? 5, 4, 3, 2, 1. Allozymes are made by different alleles of the same chromosome, while isozymes are made by different chromosomes. Next, what is the difference between coenzyme and cofactor? 5, 4, 3, 2, 1. Coenzymes are small organic molecules that help enzyme to function, while cofactors are metalines such as zinc and ferrous. Okay, next. What is the difference between apoenzyme and holoenzyme? 5, 4, 3, 2, 1. Apoenzymes are empty and holoenzymes are full of substrate and cofactor. Bingo, right? Okay. Next, what is the difference between homotropic effector and heterotropic effector? 5, 4, 3, 2, 1. Homotropic effector is substrate itself working as effector, while heterotropic effector, it has a separate molecule. Nice, right? Okay, what is the difference between competitive inhibitor and non-competitive inhibitor? 5, 4, 3, 2, 1. Okay. Competitive inhibitor take the place of substrate, while non-competitive inhibitor, it has a separate cleft in the enzyme. Thank you very much for watching that video. If you like it, please hit the button like here and share it, and please subscribe my channel. Thank you. Bye bye.